This is a presentation over glycolysis made and presented by Darren Ochoa and Trevor Hord. For Biochemistry II, Chem 4323, professor is Dr. Nick Flynn for the spring of 2017. The glycolysis metabolic pathway is divided into two phases, the preparatory phase and the payoff phase. The preparatory phase involves using ATP to prepare the glucose molecule for conversion into pyruvate in the payoff phase. We use 2 ATP in the preparatory phase and ends when fructose 1,6-bisphosphate is converted into two 3-carbon molecules, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. The first step of glycolysis is taking glucose and having it undergo a phosphoryl transfer with ATP, giving glucose 6-phosphate and ADP. A unique aspect of this reaction is that there are two isozymes that do this depending on the cell type. Glucokinase is the enzyme in the pancreas and liver, while hexokinase is the enzyme for just about everywhere else. Additionally, hexokinase requires the divalent ion magnesium to operate, and this reaction is an irreversible reaction. The next step of glycolysis is an isomerization with an isomerase of glucose 6-phosphate forming fructose 6-phosphate. This step is a reversible reaction. An important reaction in glycolysis is the PFK1 reaction, with the phosphoryl transfer of a phosphate molecule from ATP to fructose 6-phosphate to form fructose 1,6-bisphosphate. This reaction is an irreversible reaction. It's considered the pacemaker of glycolysis and is a key point in regulation, which will be touched on later. The aldol cleavage of fructose 1,6-bisphosphate into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate with the enzyme aldolase marks the end of the 6-carbon phase and the beginning of the 3-carbon phase. Interestingly enough, DHP is converted into glyceraldehyde 3-phosphate with an isomerization reaction with an isomerase. This will give two molecules of G3P to mark the end of the preparatory phase and the beginning of the payoff phase. Glyceraldehyde 3-phosphate undergoes oxidation and phosphorylation with the enzyme G3P dehydrogenase to form two molecules of 1,3-bisphosphoglycerate. Two inorganic phosphates are needed and two NAD+ are needed, which generates two NADH plus two protons. We use inorganic phosphate instead of ATP because if we use two ATP, then we would have a net gain of 0 ATP throughout the entire glycolysis process. The next step is the phosphoryl transfer of one phosphate molecule from each 1,3-bisphosphoglycerate to ADP to generate 2 ATP and 2 molecules of 3-phosphoglycerate using the enzyme phosphoglycerate kinase. This is an example of a unique method of phosphorylation called substrate level phosphorylation, which is the phosphorylation at the level of the metabolic pathway producing ATP. It is also worth mentioning that this is an irreversible reaction. Two molecules of 3-phosphoglycerate undergo a phosphoryl shift with the enzyme phosphoglycerate mutase to form a 2-phosphoglycerate. The next step is the dehydration of 2-phosphoglycerate with the enzyme enolase to form phosphoenol pyruvate. This is a high energy compound because of the enol group, giving a high phosphoryl transfer potential. The final step of glycolysis is the substrate level phosphorylation of ADP with the phosphate groups of the two molecules of phosphoenol pyruvate to produce two molecules of pyruvate and two molecules of ATP. This is an irreversible reaction. So to recap, we have the overall reaction of glucose plus 2 ADP plus 2 NAD plus 2 inorganic phosphates to form 2 pyruvates, 2 ATP plus 2 NADH plus 2 protons plus 2 waters. As seen throughout the presentation, there are 4 irreversible reactions of glycolysis. However, only 3 of them are key regulatory points. The first is the hexokinase-glucokinase reaction. Enzyme activity is based on cell type, meaning that these isozymes catalyze glycolysis depending on their location in the body. Glucokinase primarily occurs in the liver and pancreas, whereas hexokinase occurs everywhere else in the body. Additionally, these isozymes have differing Michaelis-Menten constants, with hexokinase having the lower Km, therefore having a higher affinity for glucose. This is important because that means that the liver stands back because of glucokinase's lower affinity and lets other cells have a first shot at the glucose. After everyone has their fill, glucokinase in the liver will remove glucose from the blood and convert it into glucose 6-phosphate. The second regulatory reaction in glycolysis is PFK1. PFK1 is often called the pacemaker of glycolysis and is regulated through allostery. It is inactivated by ATP and citrate and activated by AMP, ADP, and fructose 2,6-bisphosphate. When AMP levels are high, PFK1 is activated. When ATP levels are high, PFK1 is inactivated. This allows glycolysis to respond to energy requirements of the cell. The final regulatory step in glycolysis is pyruvate kinase that has a number of isozymes and is regulated by allosteric effectors. Fructose 1,6-bisphosphate is an allosteric activator. ATP, acetyl-CoA, long-chain fatty acids, and the alanine glucose cycle are all allosteric inactivators. Diseases of glycolysis are rare due to the importance in the pathway. However, it was discovered by Otto Warburg, MD, that a characteristic of cancer cells was their utilization of anaerobic glycolysis. That marks the end of our presentation. Thank you for watching.